Where is everybody? Um, I hope this isn't in response to that message I sent out that I'm recording lectures and we'll be posting that. That doesn't mean that you can stop showing up. So thank you for coming today. I'm going to be sending out an announcement reminding everyone that it's important to show up to lecture to better learn the material. I know this low attendance could also be due to the fact that you don't really have a lab today. You know that, right? So no lab session. You have that homework assignment of watching a video, and it's a really good one. Sir David Attenborough is the person doing the documentary based off his book, The Private Life of Plants. And this particular episode will be about how plants adapt to their particular habitats. And I think the video starts out with him north of the Arctic Circle and looking at plants that can handle that type of extreme. And then he goes all the way to the southern tip of Africa and all over the place. So it's a good video. So make sure to watch that as part of your lab, responsibilities. Okay. So today we need to keep going on with the gymnosperms. And I wanted to touch back on what we covered on Tuesday, looking at the idea that when you look at a pine tree, for example, which they're showing here, that's the sporophyte that you're looking at. Okay, so that's the sporophyte phase. The gametophyte phase is something that you won't see living independently from the sporophyte. Okay, it's part of the cone tissue, and whether you have the pollen cone and the female cone. We've been focusing on the female cone so far, but we'll get into the pollen cone too. And so these cones really being shown in this picture, of course, remember that, why is that doing that? That the cones can, they're attached to the tree. All right, and they stay that way, and the gametophytes will develop on the sporophyte. So we have a big shift from the previous groups of plants because the spores now will not be released like what we've been saying with the other two groups. Those spores are not released, they actually stay in the sporangium tissue. So the female megaspore is just going to stay hidden away in the sporangium tissue of the sporophyte. And then it'll develop there into the female gametophyte. And so that's a huge shift. So that's what they're showing here with the female cone. And then you can see that the female gametophyte is starting to develop. They're pointing out the eggs. So, they have the archegonia there with an egg in each archegonium. So we need to think about the evolution of the ovule. So that's the female portion. So when you zoom into the pine cone, that you're looking for the megasporangium tissue and what's happening in here. And so with the female pine cone, the megasporangium will house just one megaspore. So there's not a bunch of spores in there. They reduced it down to just one. And they call that the megaspore mother cell. And then before any of these spores in any group of plants, before they can get into the next phase, they got to go through meiosis. And so meiosis is going to happen. And when that one single megaspore mother cell goes through meiosis, you end up with 4 cells. And then we find out that 3 of those die off, and you're left with that one functional megaspore, right, through that process. And so that's being shown here, where you have the megasporangium tissue, and they talk about the megasporocyte. That's the megaspore. And I just shortened down the megaspore. And so then you can see it's going through meiosis, and the result of that is 4 megaspores, 3 die off, and you're left with that one functional megaspore. So we went through this on Tuesday. I redid these slides a little bit just to make it hopefully a little bit more streamlined. So here you're looking at that one functional megaspore. The other 3 have died off, okay? But you can see that it's still being held within the megasporangium tissue. It's protected really nicely. And then around that, you have the integument with even more protection. And so Once that functional megaspore, so now it's in the haploid phase, okay? But the megasporangium tissue that it's inside of is actually a diploid phase, right? But that functional megaspore, because it went through the meiosis process, it's half the number of chromosomes. Okay, so that functional megaspore is going to start to grow now. It's going to go through, mitosis and cell division and growth and grow and grow and grow. And it goes through this weird stage that some of the slides in the lab might be labeled the free nuclear stage. That just means it's developing, it's starting to grow and develop into the megagametophyte. Okay, so that functional megaspore is going to go through lots of growth to get you the megagametophyte. And eventually, at first, when you look at it on the microscope, it looks really odd and like there's big gaps and like dead space. And it's like, what is going on here? But eventually you start to see the megagametophyte really taking shape and everything starts to form. And then you finally get to see the archegonia showing up. And it depends on the species of gymnosperm, how many archegonia they might have. But it's the same idea. You have one egg in each archegonia. Okay, so that hasn't changed. And what you tend to see is the archegonia will develop right near the opening that is allowing for pollen to come in, right? So that kind of makes sense to put it at the end, right where the pollen's coming in, so it's easier for the sperm to find the egg. So that's the micropyle, that's the opening, which you saw in the previous picture. And so they see that pretty much all the eggs are capable of being fertilized, but it usually works out that even if they're all fertilized, only one will really make it, and the other three will arrest pretty early in development and kind of die off. And so again, it's usually only one embryo develops in that megagametophyte. Okay, so what we just described is taking this previous idea here, where that functional megaspore, you have to imagine now that megaspore going through cell division, cell division, cell division, and that free nuclear stage, and then finally forming this, where you have this kind of brown circle, and then you see these two kind of olive brown. Those are the archegonia, because it's two. And then all that olive brown, that's the big fat egg inside, right? So it's a huge whopping egg in each archegonium. And then the rest of the tissue, supporting that, that's the megagametophyte or the female gametophyte. So that functional megaspore turned into this brown area here, the female or megagametophyte with the archegonia. So that's what happened to that functional megaspore. And you can still see it's still being held within the megasporangium, which is surrounded by the integument. So you have a lot of protection for this. And so now that female gametophyte, the megagametophyte, it's like, okay, I got the eggs, everything's good to go. And now it has to hopefully be fertilized. And in order for that to happen, got to get the pollen there. And so it makes sense that the integument has a little bit of an opening to allow for the pollen to sneak in there. And what typically happens is you'll see that there'll be a liquid droplet there, and the pollen will land on that. And then as that droplet starts to kind of dehydrate, it kind of gets sucked in through the micropyle, and then it lands on the megasporangium. And so now that pollen grain essentially has delivered the sperm, now the sperm's job is to get all the way through the megasporangium somehow so that it can find the egg. So it's a little bit of a effort here that they have to accomplish. So this megagametophyte is very small, highly reduced, but it's really small. It's all held within the megasporangium. And then the integument is surrounding that. So really nicely protected. Okay, so this is all happening in the pine cone tissue, right? In the scale area. And so if you look at this entire thing, they call it the ovule, right? So this entire thing, you would say, oh, that's an ovule. Okay, and the ovule consists of this integument, the megasporangium, the megagametophyte. This is a mature ovule ready to go. So hopefully the pollen grains get in there. Once that happens, then the sperm hopefully finds the egg. That's the fertilization event. And then you end up with a zygote, which starts to develop quickly into an embryo. And it's really the baby sporophyte that it's producing, it's developing into. Okay. And again, that's all being held in the megagametophyte area that's surrounded by the megasporangium, that's surrounded by the integument. Then after all that starts happening, you start to see the shift in the ovule will start to develop that into the seed. And you'll get the production of a seed. And eventually you can see that with the naked eye. If you get a pine cone at the right stage of development and you look down on the scales of the cone, you should see two seeds laying on the surface of most of the cone scales. And they're ready to be released once they get fully mature. So this picture is part of the life cycle drawing, and they're trying to show that idea of the zygote and then the embryo. And so that's the baby sporophyte in there. It has everything. It has, the root tissue, stem tissue, the cotyledon tissue, and then eventually you'll get the seed coat developing. A little winged appendage is common so that it can flutter out of the seed, out of the female cone, and then hopefully it'll land somewhere and just germinate and grow. So this is, again, working through that process that we just took several slides to detail out. So here's that functional megaspore idea, and you have to have that functional megaspore develop into the megagametophyte. And here they're trying to show the pollination idea going on, and then finally you have the embryo that's developed. So that's the baby sporophyte in that, and then this will become the seed. If you look at it under the microscope, a real one, this is what it looks like. So here you can see the female gametophyte or the megagametophyte is in the center. Those two areas there, the dark green, those are the archegonia, this big fat egg in each one. And then the tissue around that starts here, and you can see in this area, that's the megasporangium tissue. And then the tissue around that, with that little opening, that's the integument. Then the opening is the micropyle. So I would like you to know the anatomy, okay? The anatomy of a seed, like on this slide. And also looking at, this idea with a mature ovule. I'd like you to know the anatomy and be able to identify the different parts of it, okay? Questions on the ovule or the seed idea? Okay, well, we need to shift over to what's happening with the pollen and how does that all develop. And so let's shift gears to the male cone where the pollen grains are going to be. So as you look at a gymnosperm pollen grain, what you tend to see, it consists of basically 4 cells to start with. You have these so-called 2 prothallate cells, and then you have a tube cell. They call it a tube cell because that's the one that will grow out and form this long tunnel. So the sperm has a way to get through that tunnel and get to the egg. So it's called the pollen tube. And then there's another cell called the generative cell. So that's, the generative cell is one, the tube cell is another, and then you have the 2 prothallate. So that's 4 cells make up the pollen. And usually it's in this state that pollen will get shed from the male cone. So you don't see anywhere in here yet any sperm developed yet. It hasn't happened yet. So you have the prothallate, tube, and generative cell. None of those are sperm. All right, so it gets that we call this an immature pollen grain, and that's usually how it gets shed out. All right, from the male cone. And then eventually what happens is that generative cell will divide, and one will produce this so-called stalk cell, and then the other cell will be what they call the body cell. And then one more time, you got to get that body cell to divide again, and you end up with 2 sperm. So when it's all said and done, each pollen grain should produce 2 sperm eventually. They're not there yet when they first shed out of the pollen. So this is, I believe, the image from your book. And so they're showing both the male and female development here in 2 different cones. So we're zooming in now to the male cone. And with the pollen grains, you get a lot of grains. So way more male parts, I guess, compared to what we just talked about with female, where it was reduced to 1 megaspore mother cell and all of that. So you get lots and lots of pollen grains. Those of you who have allergies to pollen, you are well aware of that. And so here you can see those pollen grains will go through meiosis, and then you have they're already showing that the pollen tube's starting to grow out, but really that doesn't happen until the pollen gets delivered to the ovule of the female cone. And so I put in this other life cycle. If this works better for your brain, feel free to use this one as well. It's the same basic idea. And again, you have the male cone here being shown with lots and lots of microspores, and those microspores will go through meiosis and develop into the pollen grains. Right? So it's the microspores instead of the megaspores. Now we're zooming into the microspores, and there's lots of them in the pollen cone, the male cone. And each of those microspores will go through meiosis, and then each of those cells can become a pollen grain. So you get lots of pollen. And then here they're showing the prothalial cells, generative cell, tube cell. So they're trying to say when it's getting shed from the pollen cone, you don't see any sperm yet. They haven't showed up yet. And it's not until they land on the ovule, then it'll kick into a little bit more cell division to get you the sperm. Okay, so if you are looking at, what is pollination in a gymnosperm, technically it's the delivery of the pollen to the ovule. That's the correct definition of pollination. It's the delivery of the pollen to the ovule. But if you were to look that up in a dictionary, they don't define it that way because they normally only focus on angiosperms, where you have a flower to think about. So no flower here, but you still have a pollination event. You have that pollen getting delivered to the ovule. So we have to kind of chunk this out and explain it in chunks, but if you actually look at the timeline of how does this all happen through a season, I'd like to walk you through that next, because it's kind of bizarre how long it can take, and then how there's things that get delayed and they're kind of offset from each other. So let me get into that a little bit. So the Pinus, that's the genus name, the Pinus, most of the conifers that genus, that's the only life cycle we're going to ask you to know in the gymnosperm realm. That's it. Just this one life cycle. We're not going to get into any of the other life cycles of the neophytes, the ginkgo, and the cycads. So just think about the Pinus one in more detail. So spring, like now, supposedly we're in spring phase of the season. That's when pollination starts to happen. So that's the immature pollen grain getting released and hopefully finding an ovule in the female cone. Once that happens, once the pollen lands onto the ovule tissue, about 1 month after that, then the female finally starts to kick in. So the female's not even ready. It still has the megaspore mother cell there. There's no egg yet. And so the pollen gets there, and it's kind of like, all right, I'm here, now do your job. And the female has to go through that megaspore mother cell has to go through meiosis, resulting in 4 cells, 3 die off. It has to do all of that with the pollen sitting there waiting. And so that functional megaspore will then divide, and become the female mega, the megagametophyte. And then that takes a while. So then 13 months after the pollen grain has already landed on the ovule, 13 months later, you finally get a more mature, starts to finalize the megagametophyte, and then that takes a couple months. So about 15 months after the pollen has landed there, you finally have the archegonium developed with an egg. So pollination happens, and then the pollen grain just sits there. Like, okay, twiddles its thumbs until the megagametophyte kicks in and does all of its development. So it's kind of weird how it's this offset, but it works. It works for them. Okay, so let's think about what's going on, because those pollen grains are still immature. Like, there's no sperm there yet. So think a little bit about that. So the pollen lands on the ovule about 3 months later. That's when you'll start to see the pollen tube elongating. That cell will start to grow long. It forms kind of a tunnel, and it has to make its way, has to kind of digest its way through the megasporangium. And then it's got to find an archegonium with an egg. That takes a while. So up to a year after pollination, then one of the pollen grain cells, the genitor cell, will go through more division and develop 2 sperm. And then about 15 months after pollination, you finally see that pollen tube has made it through the megasporangium, found an archegonium, and can discharge the sperm into the egg. So no free water necessary. It's actually the pollen tube that's really helping getting the sperm to the egg. So there's no free water necessary. One big deal that you need to think about is that 2 sperm get delivered, 1 makes it. So one of the sperm will fertilize the egg. The other sperm from that pollen grain typically dies. So 1 makes it, 1 dies. I need you to know that, because when we get into the angiosperms, we're going to say something very different at this stage. Both sperm do something in the angiosperm world, but in the gymnosperms, it's a race to the finish line. 1 makes it, the other one is done for. Okay. So if one of those sperm from that pollen grain does fertilize the egg, then you have to have more growth, of course, because that fertilized egg has to develop into the baby sporophyte. So you got lots of growth that has to happen. So they call it the free nuclear stage, because it's just a bunch of really rapid growth, but it's hard to make sense of the tissue. It looks really odd under the microscope. And then about 18 months later, you start to see a mature embryo. We have the baby sporophyte and all the seed paraphernalia that's needed to get the seed to fly out of the, float out onto the wind current out of the cone. So really, start to finish, it's usually 18 months, a year and a half, from pollination to finally seeing a seed from that. It can range, so that 14 to 20, it depends on which species you're looking at in the Pinus genus. But if you could remember 18 months, that's the number I'd like you to know. Yes. The whole previous spring, yeah. So when you're out hiking, in the fall, start looking for cones that would be, that took 18 months to show up, so a whole year back. So yeah, much slower timeline than most of the plants we've been talking about so far. So yes, yeah. Yeah, because around here anyways, they'll usually have to survive winter, right? And so if they get shed, they often, depending on when they get shed, they might have to stay dormant until spring. Yeah. And that's for this area. You know, if you're in more tropical areas or more not so long winters, they might, or mild winters. So if you look at pollen under the microscope, you can see that they also have appendages, these two things on either side. Those are kind of like air sacs, and it's just a way to keep the pollen grain. It's like wearing, you know, PFDs so you can float. But this time they're floating up in the air column. And so it's a way for them to float along the wind current so that hopefully they'll reach the female cone somewhere nearby. And so if they do, this is what you would see. This is a great picture because it's actually showing pollination occurring right there. So you have the micropyle. This is the opening called the micropyle. And then you have the integument tissue flanking on either side. And that's surrounding, this is all still megasporangium. This is the megasporangium tissue. And then this cell right in the middle here, that cell is the megaspore mother cell. So you can see when pollination has happened, there's no megagametophyte yet. That hasn't even happened. And so this megaspore mother cell has to go through meiosis. So here is another picture with it all labeled, where you can see the integument surrounding all of it, and then the megasporangium, megaspore mother cell, and then the pollen grain. And you can see that the micropyle has sealed up. So that's after the pollen gets delivered, then that cone tissue will seal up like that. And that's a good thing because it's really now quite protective. It's a nice closed, cozy environment. The sperm can hang out and wait as the megaspore mother cell does its job of going through meiosis, getting functional megaspore. The functional megaspore has to grow into the megagametophyte. And so if that starts to happen, you'll see something like this, which doesn't look like much. This is just the pre-nuclear stage. So this is the functional megaspore just going through some growth and trying to further develop into here. You're seeing finally the megagametophyte is the tissue's kind of flat, you know, filled in there. And you can see this species only has one archegonium, there's one egg in there. And you can see how that archegonium are closest to the micropyle end. That makes sense because it's less distance than for the sperm. And this is a great image because it's actually showing two pollen tubes starting to grow into the megasporangium. So that kind of blobbish look, you can see those two kind of blobs? So those are two pollen tubes. So two pollen grains landed here. Each one is sending out a pollen tube. So that pollen tube, we say it digests its way through all this megasporangium tissue. And then it has to find this archegonium with the big egg in there. So it takes a while to get that to happen. And the sperm aren't even ready yet. They're not there. But eventually, tube sperm will form, and they'll just go through that pollen tube, and no free water is necessary. This is a great image too. So the drawing on the left is mimicking what you see in the real microscope slide here. And you can see that there are two sperm that are in here. So this is an amazing microscope prep for this. This is a sperm down here. Whoops, what happened? Whoops. So right where my laser pointer is, that's the sperm. And then if you can see this little blip here on the egg nucleus, that little blip, that's the fertilization event. So they actually captured fertilization happening, sperm and egg fusing. That's pretty amazing. So this other sperm lost the race. So this one is going to die. Whereas this one made it and fertilized the egg. So you can see all of that. That's the egg. It's a big, fat, plump egg that's taking up most of the internal space of the archegonium. And so you got to get the sperm to the egg nucleus, get that fusion and fertilization. And then if that happens, so here you're looking at really right after the fertilization event. Now you got to get that developing I think I might have a better picture in a minute here. And then eventually it would look like this, where this is the baby sporophyte now in the seed. And so all this blue in the center, that's the mature embryo or the baby sporophyte. All right, and here these are the cotyledons, pine needles, the leaves. Right there, that tip right there, that's the apical meristem, your stem tissue. And then actually the root tissue, you don't need to know all the anatomy of this. I'm just, if you're curious, the root area is down here. And then all of this pink stuff surrounding it, that's the megagametophyte. So I know way back, what was it, week 1 of the semester when we looked at seeds, we talked about endosperm or cotyledons. And that was all with angiosperms. This is a gymnosperm seed. And they do not have endosperm. That's a big deal. That's a big distinction. All right. So what do they have in place of it? Instead of endosperm tissue, they have that pink, in this case it's labeled pink or stained pink. That's the megagametophyte. So now it serves the same function as the endosperm in an angiosperm because it's going to provide nutrition. So it's going to support the baby as it starts to germinate and grow into a tree. That megagametophyte tissue will deliver nutrients as it starts to germinate and grow. So the megagametophyte is a huge part of the seed and really, really important because it helps feed the developing, germinating sporophyte. So this image here labels that. So here you can see they have it nicely labeled with the different parts of the anatomy of the baby sporophyte. And then no, they don't really have that. Oh, there it is, female gametophyte. I was looking for mega. So the female gametophyte where they have that. So all the purple, in the last slide it was pink. In this slide it's purple. All that purple is the female or the megagametophyte. And it serves a function of nutrition for the baby sporophyte. Okay, so all of this is happening in the female cones. And when you look at them, they're pretty complex. They have a lot of different parts to the cone. There's some exceptions. Ginkgo biloba is very simple. I'll show you a picture. They don't have a complex like pine cone structure. They have just kind of like a single piece of tissue that all of this is happening. Very, very but believe it or not, Ginkgo biloba, which looks like an angiosperm, it's confusing. But when you look at their phylogeny and their traits, they actually fall into the gymnosperm category. And so this is a close-up of their leaves. I mean, it does not look like any gymnosperm that you usually think of. And then this is the developed ovule that will get, the egg is in there and the sperm will get there eventually. Euseeds are another idea too. Again, just really simple. Just this one little piece of tissue. It's usually very fleshy looking. And again, you would not think that, that's a gymnosperm, but it is. Okay. And then this has gone so far as the seed has already developed that little dark blob in the middle as the seed. Okay, so yes, there's some exceptions, but if you think about most of the pine cones, the female cones, they are complex strobili, really. It's, that's where the sporangium tissue is all located. So technically, it's strobili. And you can have tiny ones, like the juniper. Around here, our junipers tend to be more like bushes. They grow lower to the ground. If you go out west, I was really surprised the first time I went out west, really surprised to see juniper trees, because around here it's just bushes, very low to the ground, but They have some really amazing morphologies out west. So they have, junipers have really small strobili, very, the scales are like really tight to each other. And so when you look at them, you would say it's a berry, right? You probably heard juniper berries, right? And that's how people talk about them. And they actually turn blue. So they look like a berry, but it's not. It's a strobilus. What do we make from juniper berries? Big business. Anyone know? You're over 21, you might know. Yeah, the juniper berry, where you get that pine taste to the gin. And so it's actually juniper berries that they use in the brewing process in the distillation for the gin. But so everyone says it's a juniper berry, but really you need to know now that you're in a botany class, no, it's a cone, it's a strobila, and it's actually a gymnosperm that's producing that. Cycads have really big cones. I'll show you some pictures. It's impressive because cycads aren't that big, at least in the United States. The native cycads are down in Florida area, and they're not usually very big, but they have big strobili. Except for the cycads, it's a safe thing to say that pollen cones are always smaller. The cycads have to be the exception. But other than that, all the gymnosperms, it's the female cone that's bigger. And I'll show you pictures of some of these. And then they're woody at maturity. So the female cone is woody. And I think most of you appreciate that. You've experienced pine cones in your life by now. Okay, so let's just take a look at some of these. This is a juniper on the left, more in the tree formation rather than a bush, and then a close-up of the strobili. But it's easy to see why people call that a berry. You know, it turns blue, blue-gray. It's really easy. Here you're looking at junipers and cypress. Again, really small cones. The scales are very tight together, all kind of fused into this roundish shape. Cycads, on the other hand, the male cone is the taller one here, okay? And then the female cone is usually fatter, wider, but shorter. And if you weigh it, the male cone wins in weight. So they say the male cone is bigger in the cycads, but in the all the other groups, it's the female cone. So if we look at a male cone of a conifer, for example, so that this is why they call it a strobulus, because when we were talking about that in the previous groups, a strobulus is just a dedicated area where you have all the sporangium tissue clustered together. And so that's what you're looking at. Each of these is sporangium tissue, and all that grainy look inside, those are the microspores. So you can see a pollen cone is producing a lot of microspores. And each and every one of those microspores can develop into a pollen grain. Actually, each of those microspores has to go through meiosis and produce 4 cells, and then each of those cells develops into a pollen grain. So lots of pollen. Now you can appreciate why there's so much pollen in the spring. So what's going on inside that male cone is that you get, again, this microspore mother cell, but there's lots of them. It's not just reduced to one, like what we saw in the female. The male goes for quantity. And so you have the microspore mother cells. Each one of those goes through meiosis to produce 4. And then each one of those can develop into a pollen grain. So lots of pollen. So there's no reduction in the microspore mother cells. That does not get reduced to 1 like we saw in the female case. They just keep it up, everything. Keep all the microspore mother cells. Each one of those goes through meiosis, and then you get a bunch of microspores. Each one of those starts to develop into a pollen grain. And then when it's all said and done, right as it's starting to leave the pollen cone, it will consist of 4 cells. And that's going to be different when we look at the angiosperm. We're going to see a difference. and that it won't be 4 cells, it'll be 2. But with the gymnosperms, they do 4, and then eventually, so you've seen this. So essentially the pollen grain, yeah, you could call it a microgametophyte. It's when it's shed, it's still immature, but technically it's the microgametophyte of the gymnosperms. And once it's shed and once it lands on the ovule in the female cone, then eventually you'll get the sperm to show up. And the sperm have to just kind of go through that pollen tube. They don't swim in water like all the bryophytes, and you see this basket. You don't have that. So the pollen grains, again, under the microscope, you can see those air sacs. And then what they did is they put a chemical on the pollen grains to get the pollen tube to start growing. And so they're showing the stage where the pollen tubes are starting to come out. And then this is under the microscope would be the female, is part of the female. So you can see it's different in appearance compared to the male cone. And it's quite woody. And you have to really zoom in to the top. So each one of these is a scale. So what you're doing here is looking at, so that's a scale where can you see the red outline I just drew? So that's a scale of the female cone. And where you need to zoom in on each of these scales, some are easier to see than others. Do you see there's a difference right there? It might be hard to see where you're sitting. That's the ovule that's sitting there on the top of the scale cone. So that's the ovule that needs to go through all that development into the megagametophyte. So the female cone is definitely more complex in structure than the male cone. And because of the way that previous image, we only saw one ovule, but you have to appreciate when you look down onto the surface of a scale, there's actually 2 ovules per scale. And each of those ovules, if the egg gets fertilized, each of those can develop into a seed. So if everything went really well in that pine cone, the female cone, when you look down on it 18 months later, technically, hopefully you see 2 seeds on each scale of the cone. You'll see 2. And then there's usually this, you don't need to know the subtending bract, but it's just more complex. I'm just trying to point out that the female cone is more complex. There's more parts to it. And the scales are usually arranged. If you look down on it, it's usually a spiral arrangement. So it's complex. And then what you tend to see, look for this screen here, you'll see some cones on a tree with the scales open. And you'll see other cones on the same tree where the scales are fused. And I remember when I first saw that when I was, back in middle school, it's like, weird, why they do that? But you have to remember, they're going to be in different phases of the life cycle. So some cones are ready to accept pollen. And so it makes sense that they put their scales wide for the pollen. And then if pollen has shown up and actually pollinated, then they close up to protect. So that's one thing to look at. Are hiking around, tell what phase of life they're in. Are the cones open, the scales open, or are they closed up? And so once pollination happens, then that's when you start to see things that are. Okay, so the female cone typically almost always bigger, except for the cycad, more complex, and each scale should have 2 ovules, and hopefully 2 seeds will develop from those ovules. So here again, I took that previous image and zoomed in a little bit. So again, you can't see both ovules in this because they sliced down the length of the cone, so you can't see that. But you have to imagine another one is behind the ovule that we can see here. So on this image, you can see where I'm circling, those are ovules on top of the scale. So it's in that region that you have the megaspore mother cell, has to go through meiosis. So after you have that egg produced, you get fertilization, and then you start getting the seed to develop. What they find is that the integument tissue that's surrounding and protecting, that actually morphs into what we call the seed coat of the seed. So when you dissect a seed coat or a seed, the seed coat, the outermost portion of the seed, that's technically integument tissue that we've been looking at in a lot of these images. And then there's no endosperm inside the seed. There's not that, but there is the megagametophyte. That serves the same function as the endosperm to provide nutrients. And so then after you get full seed maturation, everything's good to go, then the scales will come loose again from each other and will spread out. And then that's when you can see, oh, look at that, there's 2 seeds on each scale with that little winged appendage. And then hopefully a brisk wind will take those seeds and let them disperse. There are, you probably learned in some ecology class at some point, there are pine cones that won't open, even though the seeds are good to go. They won't open. And that's an exception to the rule. Really, there's only a few that do that. And they need the extreme heat, so they need a fire to sweep through that area. And the fire actually triggers the cones to open their scales. And so it's just a strategy to repopulate the area with the seeds and after it's been cleared by the fire. So lots of different, as you saw in lab, we had a bunch of pine cones for you to look at. I know Emily was asking, we have a Coulter pine cone that's massive and super heavy, and she couldn't find it. So I dug through, someone had stuffed it away, probably because they just wanted it secured. So I went and grabbed it. So you'll see that next week in lab, but it's called the Widowmaker pine cone because it's so heavy. And it's from a Coulter pine tree out west. And you have to wear a hard hat if you're in those areas. And even that might not totally protect you. It has caused death when it falls off the tree and hits somebody. So it's mad. I don't even like holding it because it hurts my hands because the sharp points of the scales kind of poke into it. So I could definitely see it, and it's heavy. I could definitely see why they call it the Widowmaker. So one thing to think of too, if when you're out hiking, you're looking, often you'll find the male cone and the female cone on the same tree. But what tends to happen is the male cones are lower on the tree and the female cones are way up top. So look for that when you're out hiking. And of course, size difference, so the male cones are always small. But it makes sense for a tree to do that because if you're in a forested system, putting your pine cones down a little bit lower prevents self-pollination. Because I think if you've been hiking in the woods before, very rarely total straight updraft of wind, it doesn't happen. You have wind, that comes from east or west or south, but you don't have these drifts. And so that makes sense. If you put your pollen down low, there's no very low chance of updraft is going to happen in the forest to cause pollen of the same tree. So it's a way to cross-fertilize, get the pollen from a neighboring tree. Okay. All right, we'll keep going with cones on next week and finish up the gymnosperms, or at least the conifers, and then we have to talk about the other 3 groups of gymnosperms.